We will be discussing about motifs. So what is the meaning of the motif? Motif is nothing but a part of a protein. Let's say this is a double-stranded DNA and this is some protein. Now motif is a particular part of a protein which interacts with the DNA. The remaining part of the protein, like this one, may as act for several functions which we will discuss in just a minute. So now you are clear that this is a definition in the Harper that motif is that part of a protein that makes direct contact with the DNA. So what does the other part of the protein do? It may act as trans elements which can act as heterodimeric area. It may provide surface for the heterodimerization. It may also provide surface for the dimerization. It may also provide the surface for the interaction with the other coactivators, repressors. So this is about a little brief about what motif is. We will discuss motifs in great detail now. We will be discussing three types of motifs, that is helix, turn, helix motif, zinc finger motif, and leucine zipper motif. These three motifs we will be discussing in great detail now. So we will first take up the helix turn helix motif. In the helix turn helix motif, it is a dimeric protein in which each monomer has 3 alpha and 3 beta chains of proteins. Now these 3 alpha and 3 beta chains are arranged somewhat like, let this be one alpha chain, this be the second alpha chain, this be the third alpha chain, and this is the one beta chain, second beta chain, third beta chain. Similarly, in the dimeric form, the other part will look like one beta chain, its second beta chain, third beta chain, and similarly the one alpha chain of second monomer, second and third alpha chains. So this is somewhat like helix turn helix look likes. There is one side the presence of, this is one monomer, this, and this is next monomer. It is the dimeric protein. Helix turn helix motif is a dimeric protein in which there are 3 alpha and 3 beta in one monomer and 3 alpha and 3 beta in second monomer. This constitutes the helix turn helix motif. We need not to go into the details of the structure. We just know something about that. Just remember that it is a protein having dimeric protein having 3 alpha and 3 beta chains in each monomer. So it look like this, that you can see here there is one alpha chain here, one alpha chain here, one alpha chain here, and one beta chain here, one beta chain here, and one beta chain here. So this is one monomer and this is another monomer with alpha and the beta chains. Now we will see what does motifs do, what is the role of, you can say, motifs करते क्या है हमारे जीनोम के अंदर. So दे हैव रेगुलेटरी रोल, जैसे कि फॉर एग्जांपल हेलिक्स टर्न, हेलिक्स जो मोटिफ है ये क्या करता है, इट रेगुलेट्स डेवलपमेंट ऑफ स्ट्रक्चर्स ड्यूरिंग फीटल स्टेज, इट इज़ एम्ब्रियोनिक डेवलपमेंट में हेलिक्स टर्न, हेलिक्स मोटिफ रोल प्ले करता है। ठीक है, फर्दर हम रोल्स बाद में डिस्कस करेंगे जैसे जैसे वी विल डिस्कस द नेक्स्ट टाइप ऑफ मोटिव्स. नाउ वी विल टेक अप द जिंक फिंगर मोटिव. Before that, just for the information, note that the helix turn helix motif was the first motif to be identified, and the first described motif was the helix turn helix motif. Now, what is in the zinc finger motif? In the zinc finger motif, there is something a finger-like structure having zinc at the core, bounded by, this is particularly a chain of, you can say, protein chain. It's a protein chain having various molecules, various atoms. Here are the atoms of the carbon or hydrogen. There may be hydrogen atoms here, and zinc is attached to these elements via covalent linking. So this is the structure of zinc finger motif. Zinc finger motif has a role in intracellular receptor to which steroid hormone binds. You might have listened about the hormone receptor elements in the DNA. उनमें जो ये होता है जिंक फिंगर मोटिफ, उसकी रेगुलेशन में हेल्प करते हैं जिंक फिंगर मोटिव्स. अब कैसे अटैच होते हैं? लेट दिस बी द DNA. So let this be the minor groove, this be the major groove. In the major groove, this zinc finger motif attaches like this and control the hormone response elements. This is a zinc here. Control the hormone response elements, the molecular mechanism of which is not in the UG curriculum. Now we will discuss about the leucine zipper motif. Now what is the leucine zipper motif? So इसमें क्या होता है, ये एक प्रोटीन मॉलिक्यूल होता है जिसमें एक के ऊपर एक कॉइलिंग है. दिस इज़ नॉट अ 2D structure, it's a 3D structure. If we see in a side view, it would look like this. So ये something like it's something like helical turns, जिसमें like you can say solenoid होता है, उसी तरीके से होता है ये, something like this. इसमें क्या होता है, after every 8 helical turns, sorry, इसमें total 8 helical turns होती हैं, जिसमें every 7th पोजीशन पे ल्यूसिन होता है. जैसे कि एक हेलिकल टर्न है, लेट दिस बी द 1 हेलिकल टर्न. हर हेलिकल टर्न में 4 अमीनो एसिड्स होंगे. सो आफ्टर एवरी 4 in each turn there are 4 amino acids. So leucine comes after every 7. So it is like 1, 2, 3 and 4. Let this be the 1 turn. After next turn 5, 6, 7. So here will be leucine. Next again 2 टर्न्स होंगी, फिर ल्यूसिन. इस तरीके से एक हेलिकल स्ट्रक्चर बन जाता है जिसमें आफ्टर एवरी 7th पोजीशन और एट एवरी 7th पोजीशन देयर विल बी द ल्यूसिन, एंड देयर 8 हेलिकल टर्न्स इन टोटल. सो कुछ इस तरीके का स्ट्रक्चर बनेगा जिसमें हर कॉन्जिक्यूटिव 7th पोजीशंस पे क्या होंगे, ल्यूसिन. तो इस टाइप के मॉडल को बोलते हैं ल्यूसिन जिपर मोटिफ. So in total there are 8 helical turns and there are 4 amino acids in 1 turn. So 8 times 4, there are 32 amino acids in total in one leucine zipper motif. So at every seventh position there will be a leucine. About 4 leucine are there in one leucine zipper motif. So work, you can say the function of the leucine zipper model, leucine zipper motif is regulation of growth regulators and oncogenes. So basically what you have to remember is that in the zinc finger motif there is regulation of the hormone response elements, specifically steroidal or the thyroid hormone regulation. In case of leucine zipper motifs, there is a, regulation of oncogenes and growth regulators. In case of helix turn helix motif, there is regulation of structures of embryonic development.